Okay, fantastic. Right. So it's just turned 10 p.m. where I am here in Chengdu in China, where we have it's a very a busy time over here. We have a parallel sort of a conference taking place, um, which has been sort of fascinating all around sort of EEG and the next 100 years. And delighted to have a sort of specific session dedicated to this sort of topic with two fantastic sort of speakers alongside me in Professor Babiloni and Professor Schalk. Um, I will kick off this symposium. I will talk to you about some recent work that we did on this very topic and about what needs to be done next to help promote, at least in our perspective, the sustainable growth of EEG. So I will start sharing my slides now, and hopefully you can see them. Okay. And if I don't hear anything from you, then I will assume that that is all okay. And I will also just get going with our
Yes,
timer to make.
it's fine.
Yes, excellent, fantastic. Thank you very much for confirming. Okay, so right. We've heard this sort of whole conference is dedicated around the sort of EEG 100 years. You also heard from Professor Rossini just now about the sort of last 100 years of EEG for clinical practice, which is very comprehensive, and then some future sort of avenues. And so I'll try not to sort of go over old ground too much. You've already seen this slide if you joined yesterday from Pedro Valdez Sosa. And all I will do here is just flag this portrait on the left-hand side here, which is created by a PhD student at McGill University, which is actually just a combination of sort of waveforms to create the profile of Hans Berger. And in fact, if you would like to sort of try out and recreate some of EEG sort of historic sort of figures, you can download the script and create your own portraits. It's actually quite an impressive piece of work from Charlotte Maschke. So thank you, Charlotte, for submitting that. Now what I want to show you, and this I would like to call this a world exclusive, but actually I showed this at the EEGNet conference a few months back. And so if you didn't see it, it's hopefully going to be entertaining for you. And if you did see it, I think it's worth another watch. So what is this? So we've heard about the discovery, Hans Berger, but you may not know exactly how it was done. And so National Geographic, actually in France, went through the pains of recreating exactly that moment in time. And I'd love to sort of just share this with you. So there is no, I've turned the audio off. Sorry for people who are native French speakers, you will benefit from the audio version, which I will share happily afterwards. But let's take a moment just to watch the discovery of EEG. So that is it. That is it's always amuses me every time I watch that video. It's sort of the birth of a sort of supervillain, perhaps. But that's the sort of fantastical sort of story about Hans Berger. Of course, not exactly true to reality. What is true is that it really did capture the imagination, this discovery, sort of described as a sensational one in the German press in summer of 1930. And what I want to do is to extract a few excerpts from a book by Cornelius Bork, really fantastic sort of history on the cultural side of EEG, how it's influenced us over the sort of the years. And I want to just pull out some examples. I'm not going to read all of them, but you can see here that sort of what happened after that discovery is people wrote into Berger or they wrote into the newspapers reporting on this sort of work on all the possibilities of EEG. Right. So from measuring behavior in the head and during pregnancy, on mood, providing some kind of objective measurement of mood. And in fact, also in terms of measuring the temperament of a horse, which I found really sort of fascinating. through to sort of comparing sort of different mental processes, in fact, motivated by demonstrating the utility of one sort of calculator product over another. And then through to sort of chair manufacturers identifying the comfort degree of a chair and trials on sort of different types of sort of coffee. So really, so what you can see here is there's so many different possibilities opened up with this brain script, right? This excitement around its potential, all these differences. So from telepathy, the comfort of the chair, as I just mentioned, through to the measurement of the temperament of a horse. And this brain script is what it was described as. And of course, we've already heard from Professor Rossini, I won't sort of labor the point again, is that what it required before we were able to make any moves and any progress in any domain of sort of the application of EEG, we required some robust replication of these sort of phenomena. Really needed to sort of provide a degree of sort of trust for the scientific community to start adopting it. And that was provided here by Adrian and Matthews and sort of Lord Adrian, sort of a Nobel Prize winner, was a driving force behind much of sort of a discovery in neuroscience in those years. And then here's again some excerpts from their work. They found it difficult to accept Berger's observation, so they decided to repeat his experiments so that they could be satisfied. And the fact that they were able to demonstrate this really lit the sort of the torch for subsequent sort of applications. And there are numerous discoveries following that. You've heard some of them today, yesterday, of course, with the sort of the Irwin Roy John sort of symposium, some incredible sort of efforts to map on EEG to various different sort of phenomena. And this is a sort of timeline here created by a colleague of ours in Estonia, Dr. Richard Nahr. He has an interactive sort of timeline, which you can sort of access through that link and all that sort of QR code, so you can see some of the early discoveries of sort of EEG. So where are we now? So what do we have? Well, so horses seem to be sort of a solved sort of problem. I was really pleased to see this A&T Neuro sort of advert here on sort of EEG for horses, really sort of fantastical work there. And there's also some other stuff out there in the literature as well on designing the EEG sort of system to measure EEG in horses. So that seems to be a solved issue. Other sort of applications like the sort of monitoring sort of the chair and the calculator that I highlighted earlier. Well, neuromarketing, this seems to be making good progress as well. This is actually quite old work that I was sort of involved in many years ago now, but there's been a lot of progress in this in terms of characterizing the neurocorrelates for making decisions on whether to purchase or rate the preference of products. And so when this sort of anniversary started to come around, what we wanted to do as sort of a community of sort of people related to EEGNet, Global Brain Consortium, and EEG Mini Labs. These are sort of all international initiatives that you've either heard of over the last couple of days or maybe hearing about in the sort of coming sessions. Is we came together and we said, okay, well, how can we mark this sort of seminal moment in time, the same way that sort of all those sort of amazing ideas were sort of captured in 1930, following the publication in 1929. And so we basically wanted to create a time capsule of what sort of neuroscientists believed the impact of EEG had been, and also what the sort of potential for EEG could be. And so we distributed a survey. I'm sure many of you were involved in sort of the collection in some of the sort of responses, completing some of those sort of surveys. And we ended up with about over 500 experts from over 50 countries, and collectively 6,000 years of sort of a EEG experience. And so hopefully the responses there in their sort of in that form have some utility and can guide us in the sort of future. But what we do know is that any kind of prediction, especially when you start to project that far out, is almost inevitably going to be wrong. And so what we are hopeful of is that the ways in which it is wrong will be interesting and sort of guide future sort of efforts. And so we have we know this, of course, from the predictions of AI and general AI and various other sort of prophecies about the technology. And so here, I just want to highlight some of our sort of predictions. We asked people, we made a bunch of statements assisted by AI, as it happens, actually. And we said, okay, well, when do you think this thing might become widely accepted amongst the sort of scientific community? And I'm just going to flag a couple of these, given that we are at the Global Brain Consortium General Assembly, just to give you a projection of where we are, how far away we might be. And so it's interesting, we often talk about EEG. In fact, just last mentioned in the last talk about it's relatively accessible and affordable, but perhaps not in every part of the globe quite yet is the sort of is the response coming back from the sort of community, with this rough estimate about it being about sort of 25, maybe 25, 30 years away for it to be accessible in every part of the globe. And then subsequently being used globally as an indicator of brain health is, again, we are maybe a few decades away from this being a reality. We also took the opportunity, we want to do some sort of serious work, not just sort of some fun sort of projections about the future. We also said, okay, well, what changes sort of would really drastically improve your working life as a neuroscientist? Where should the community be concentrating its efforts? And so we ended up with this sort of response. We came up a bunch of sort of domains of sort of activity, and we said, okay, well, how high, how important is this to your work? And we end up with this this plot here, which shows that actually people think most of these things are important. It's just a relative prioritization sort of issue. I find that artifact detection and removal methods are important. Standardization is critical, as well as sort of the development of portable and wearable sort of EEG systems and so on. I won't read the whole thing out for you. You can sort of take a look for yourself. And the question is, how are we going to start realizing these priorities? So what we did once we had these data is we started coming up with a set of principles. So a sort of a proto manifesto in some way. Really, we said, okay, well, if we are going to really achieve these ambitions, these bold ambitions, and perhaps even drastically reduce the time that it takes to deliver on some of those sort of goals, we need to implement 3 principles as a sort of community. The first is to focus on validity. We need to ensure that our work is robust, reliable, and replicable in the same way that sort of Adrian and Matthews ended up replicating sort of Berger's work. We need to be able to we need to have some trust in our sort of research and our outcomes, so that other people can build on those. We also, that democratization is extremely important. We need to have a diversity of data. What we know is that a sort of extremely large percentage of the data that we have on the brain comes from a very small sort of a proportion of the global population. And we think that's problematic. I'll explain in the next couple of slides. And then finally, is a responsibility, and there's a responsibility in all of us, really, to consider some of the issues of equity, access, privacy, and then sort of ensure that we do our part in trying to sort of make this technology as sustainable as possible. I'm mindful of time, so I'm going to skip sort of very quickly over some of these sort of points. But validity is something that's being tackled actively right now. Project EEG Main Labs, I just briefly mentioned it earlier, led by my colleague, Dr. Yuri Pavlov in Germany. And he has been coordinating an international sort of program of work where we are replicating sort of 20 of the most influential studies ever published in EEG science. And the idea is that we hopefully will be able to build a firmer foundation on some fundamental sort of EEG phenomena as a result of that. And you see there's a sort of a global effort, but you'll also see that there are lots of white spaces on that map on the top right. And so we have there's some work to do here in terms of increasing our engagement distribution across the sort of the globe for it to be a truly global effort. And we welcome your sort of involvement. So if you're interested, if it's the first time that you've heard of this and you'd like to learn more, then there's a sort of a QR code there. And if you search for EEG Main Labs, I'm sure you'll be able to find this sort of a project. I mentioned democratization and sort of diversity associated with that. And we've heard about sort of dementia and MCI recently and sort of and so one of the issues, again, I pointed to the fact that we have only a very sort of biased sort of a collective data set in neuroscience. And on the left-hand side, here is some work showing that this is an extremely big problem in genetics. GWAS sort of research here, you'll see in these panels A, that's panel A, the sort of distribution of sort of data across different sort of populations. And on panel B there, you see the sort of predictive accuracy. And basically what it shows is that, well, if you don't have much data on a particular population, then your predictions are not so good. And if we really want to have a particularly sort of a representative sort of neuroscience, then we really need to do much more in terms of collecting data that is representative of the global population. And that is definitely one of the sort of the goals of the Global Brain Consortium. And to make that a bit more concrete, here are a couple of examples of sort of initiatives that are trying to drive such work forward. So actually a couple of years ago now, we sort of published a sort of white paper on the challenges and prospects for running longitudinal studies on children and adolescents to examine adolescent development. And so that's an area that we are actively sort of working on sort of putting together. And there's also some work that came out sort of a couple of weeks ago, really beautiful work here showing the sort of a diversity in sort of brain health as measured by these sort of brain clocks. And that's my coming to the end of my my talk. That's an example of some really excellent work there, sort of published by Agustin Ibanez's team. And then finally, mention responsibility, collective responsibilities. We all have a role here to sort of play in the future of EEG, whatever that might be in methods development, tech development, in empirical research, or whatever that is. And so I just want to highlight some work here by sort of Max and Alex, people sort of key to the Cutting EEG sort of conference, who have really helped sort of make explicit what these responsibilities might be. And they will be sort of launching a sort of a manifesto for EEG for the next 100 years at the Cutting EEG conference. And that's also sort of a hybrid sort of a conference. So if you're able to join that, then please do. And what they are putting forwards is an online platform where you can make a public pledge in whatever domain of EEG that you have your expertise to help support the growth and sustainability of this sort of a neuroimaging sort of tool deep into the 22nd century. So that's a really, I think, important piece of work for helping us get to some of those priorities and some of those sort of ambitions for EEG. So I am going to close here. If you want to sort of read more about this sort of work, then there's a QR code. It's also just eeg100.org. As I said, thank you for listening, and thank you to all of our sort of survey respondents and all of our sort of co-authors who help support this sort of work. So that is, I will stop sharing my screen. If there are any questions, I'll be happy to sort of answer those. I'll give a moment if you'd like to sort of, yes, Claudio, is that a question? Yes.
Yeah, thanks for your presentation. It was a fantastic survey. My question is about your interpretation of the fact that EEG experts stated that the hardware for EEG recordings for non-users is a low priority, even if this can open a new window in the monitoring of brain function, for example, in older people at risk of brain diseases, aging diseases, and so on.
Yes, that's a really fantastic point. In fact, that's actually something I was a little bit disappointed to see that rank so low myself, actually, because I feel that there's a lot of utility in creating these extremely user-friendly devices. It would just open up their sort of application in so many different ways. But I think it does reflect the fact that there are still numerous sort of technical challenges that people believe, particularly in the sort of laboratory-based sort of research, but also this, Claudio, might be a reflection of the sort of people who are responding on their own sort of areas of interest. And so we didn't really dive too deeply into this because, to be honest, the richness of the information, we would be here forever. So what we have done is we've made available all those data, and people can sort of stratify sort of responses accordingly, and we can sort of see. Actually, I'll take a look at this sort of afterwards as well. We'll see whether that varies depending on whether you are clinically oriented, whether you are doing basic research, whether you're methods and so on. So we can see whether that sort of priority shifts according to that. But it's a really interesting sort of observation, and I agree with you. I think this is it's an important sort of a milestone for us to sort of get to.
Thank you.
Okay, so we will, I think there's any other questions there, so we'll move on to the next talk, which actually is from Claudio, Professor Babiloni from Italy, who's going to be talking to us about Hans Berger's dream. So intriguing title. So I'm going to hand over to you, Claudio, and so you can take it away. Yeah. Thank you.
Did you see my slide?
Yes, we can see it perfectly. Thank you.
Okay, yes. Thanks for inviting me to this session and Global Brain Consortium annual meeting. And you see here we are the co-leader of Global Brain Consortium Dementia Work Group. And so this is an outcome of me and Gosha Viana. And you see today that Hans Berger, a German psychiatrist, first discovered human EEG in 1924 and was the first to name alpha and beta waves from resting state, eyes closed EEG, and was the first to put in relation resting state EEG with vigilance states and also pathological brain conditions such as dementia, for example. And this is the first clear EEG recording from just one bipolar channel, frontal and occipital, from the scalp. And you can see that he was the first to describe this oscillation of EEG activity about 10 cycles for 1 second, the alpha rims during eyes closed condition, and the disappearance of this alpha wave during eyes open condition, but also in other experiments during stimulation and cognitive tasks. And so the hypothesis of my talk and our work group is that EEG in the resting state condition, eyes closed and eyes open, is a window on the neural system related to vigilance, consciousness level. And you see that converging studies show that these alpha rims are related to ascending reticular formation from brainstem, basal forebrain, in the modulation of thalamocortical circuits, and so changing the arousal of the cerebral cortex in the generation of EEG in the resting state condition. And you see in the picture the vigilance with some mental fog, brain fog, some disruption of vigilance in some patients with brain diseases. And so we want to apply these EEG recordings in patients, older patients with dementia. And you see that dementia is a clinical condition with severe cognitive deficits and disabilities in the daily activities, such as washing ourselves or prepare meals or shopping and so on, needing a caregiver to support you in these activities. And you see on the right side some pathologies, brain pathologies related to accumulation of proteins in the brain, amyloid beta and tau in Alzheimer's disease, and Lewy bodies due to alpha-synuclein in the Parkinson's disease, Lewy body dementia. And these are the main aging diseases related to neurodegenerative progressive processes in the brain. And this is the presentation of our dementia work group in Global Brain Consortium. And so I will revise quickly some findings from international consortia, global consortia, collecting data from several clinical units in several countries. And this is the European and Asian PD Waves Consortium. And you can know more about this consortium from this website. You see some key researchers here. And the PDWA consortium replicated the resting state, eyes closed and eyes open condition in participants, older people, healthy from a cognitive point of view, in Alzheimer's disease patients, Parkinson's disease patients, and Lewy body disease patients. Here you see one finding showing in the healthy older subjects a very classical power spectrum in the sources. You see in the vertical axis the cortical activation in the sources for delta, theta, alpha, beta, and gamma frequencies in the horizontal line, and 3 cortical regions of interest in the source space, Iloreta source space. And you see that in blue you have a highest closed condition with a peak of activity in alpha in the parietal occipital areas during eyes closed, and a big drop of alpha as expected during eyes open in red. These are group data. And on the right side, you see the same conditions in Alzheimer's disease with dementia patients. And you see lower alpha power peak in the posterior areas, in the eyes closed condition in blue, and a drop, but not so big as normal subjects during eyes open condition in red. And so you can compute the percentage of this kind of drop of alpha power density during eyes open condition in contrast to eyes closed condition. And so you can here in this left graph, any dot is a person, and you see that most of healthy subjects in blue showed, a lot of participants show this percentage reduction of alpha power activity during eyes open. You see negative values showing this reduction of alpha. But a lot of Alzheimer's disease patients in red did not show this kind of reactivity, showing this vigilance disorder in Alzheimer's disease patients. And the same is true in patients with dementia with Lewy body, DLB in green. So you see that is really important to emphasize the vigilance consciousness disorders in Alzheimer's disease patients and the dementia with Lewy body. And the right, you see that a big percentage reduction of alpha in central parietal occipital cortex in blue in healthy subjects, and a limited reduction of alpha in Alzheimer's disease and Lewy body disease. And exactly the same finding, the work group, the international work group, obtained in Parkinson's disease dementia. You see in green that a lot of patients with Parkinson's disease did not show reduction of alpha during eyes opening. And the same, it appears when you compute the mean percentage reduction of alpha during eyes open in contrast to eyes closed. You see the upper line, Parkinson's disease with dementia, showing less percentage reduction of alpha during eyes open. And this is another very active global consortium working on dementia, the Latin American Caribbean Consortium on Dementia, led by Agustin Ibanez and Mario Parra. You can follow Mario Parra's findings in the next symposium. They collected a lot of data from several Latin American clinical units and also non-Latin American clinical units. And you see more than 1,000 EEG recordings for these studies. And the first finding I present you is related to the association between the Berger's alpha rims in eyes-closed condition and several important variables, showing the importance of alpha rims in the resting state recording to investigate brain changes during aging. You can see in the graphs on the right side that there was a strong association between alpha power in occipital and parietal regions of the source space. You see in green the cortical regions of interest, and age as the most important association, so sensitive to aging, and sensitive to cognitive status of the persons. You see that in the frontal lobe, the alpha power and frequency is sensitive to education attainment. And so you can have a really sensitive alpha measure of the brain state. And in another study of the consortium, they were able to collect data from more Latin American and Caribbean countries and more non-Latin American and Caribbean countries for more than 5,000 participants, mapping all our planet. And they use complex and advanced procedure to analyze all these data. And you see also not only power density, but also functional connectivity measures and graph measures. And they were able to use machine learning tools from Western country EEG data to train these tools to predict the age of the person. And so when they train these machine learning tools, they use these tools to predict the age of persons from Latin American and Caribbean countries based on EEG. And so the discovery is that if you see the graph, upper graph, healthy control, non-Latin American countries, the prediction was, of course, fine. So the EEG and the machine learning tools were able to predict exactly the age of the person. But when they use this tool to predict the age in persons from healthy control persons from Latin American and Caribbean countries, it appears that the machine added some years to these people. And so they concluded that persons living in Latin American, Caribbean countries have some accelerated brain aging, and so more prone to neurodegenerative diseases related to age. And you see in the right part of the graph that the machine learning tool added some years to even more to mild cognitive impairment patients and Alzheimer's disease patients. So for another outcome of this work is that mild cognitive impairment and Alzheimer's disease can be interpreted as a hyperacceleration of brain aging in these persons. And another outcome of the work was that females from Latin American countries and Caribbean countries show even more acceleration of brain aging. And you see that the machine learning tool predicted even more years in females than males from Latin American countries. And the same was true for Alzheimer's disease patients. So the females are subjected to a more quick brain aging, probably due to less diagnosis, less therapies during the lifespan, worst nutrition, and less physical activities, and so on. So there is a social and economic factor accelerating the brain aging in Latin American countries, especially in females. Another global group was from Alzheimer's Association. And you see that the recent outcome was a review of the literature to demonstrate that COVID-19 brain fog, and so people after the positivation of and so no more virus, they still have some difficulties in concentrating, some mental fog, in some cases also some cognitive deficits. And so in the review, the work group, the global work group from Alzheimer's Association discovered that you have the same or similar effects from amyloidosis, brain amyloidosis in Alzheimer's disease, and the neuroinflammatory processes induced by COVID-19 at the level of microglia, astrocytes, and you can find even similar changes in the EEG in Alzheimer's disease patients and people with COVID-19 and brain fog. Reduction of alpha, increase of delta. So one can predict that people developing Alzheimer's disease and having some brain fog post-COVID symptoms may have really an increased risk of a fast disease progression. And so I can conclude that in the panel of biomarkers for Alzheimer's disease, for example, sampling cerebrospinal fluid and measuring a beta amyloid or phospho-tau, or with PET, amyloid PET or tau PET, and structural magnetic resonance imaging for atrophy and neurodegeneration, one should use resting state EEG, the Berger's resting state, eyes closed and eyes open EEG, to map the abnormalities in the ascending system regulating vigilance. And this is a typical symptom, this abnormality with sleepiness, drowsiness, in the patients with Alzheimer's disease during daytime, even in the morning. Thanks for your attention.
Thank you, Claudio. It's a fascinating talk. Are there any questions? If anybody would like to raise their hand, then we can take, we have a minute or two for questions. Claudio, I have I don't know if you'll be able to answer this. A difficult sort of question is, if Hans Berger was here today, what would you think he would make of the situation of EEG in a general sort of sense? I know you, as an approximation.
I think that he would be not very happy, because he predicted a penetration of EEG techniques in the clinical practice of psychiatry and neurology and or dementia, and we know that unfortunately just for example, epilepsy and consciousness disorder, coma, persistent vegetative states, or anesthesia, for example, are the domains with current use of EEG. But until now, not in the clinical practice of dementia, Alzheimer's disease, Parkinson's disease, so on. but we can try to reach the Hans-Berger's dream as soon as possible.
Mm-hmm. Mm-hmm. Thank you very much for that. Thanks for that. That speculation is a difficult question, I know. And so on to Professor Gerwin Schalk, who is at Fudan University, and he'll be talking to us about electrical brain recordings and going from simple visual observations to detailed insights into brain function. So over to you, Professor Schalk.
Does everyone see my screen?
Yes, we can see that. Thank you. Yes, perfect.
Okay. Thank you very much, Faisal, Pedro, the whole team, and organization team, a lot of who are in Chengdu. Sorry I can't be there, but it's really an impressive group of people that you've brought together, and it's a really wonderful conference. So today I'll also start with Hans Berger, but before I'll get there, I'll ask everybody if they know this man. He was actually mentioned earlier today. This is actually Herb Jasper, one of basically contemporary to Hans Berger, who sent a Christmas card to Hans Berger in 1938. So this is the Christmas card, and you can see Herb Jasper right there. He was a famous neurologist at the Montreal Neurological Institute, and you can see electrodes on Herb Jasper's head, making up these alpha waves that, of course, we've heard about. And you can see how the alpha waves make up some message, wishing Hans Berger a Happy New Year, and so forth. And so that really represents the sort of this dream that maybe at some point it may be possible that we can understand the EEG, and it will tell us important things about that person. And I've heard a lot of, in a way, frustration by people that we haven't really learned that much, but I'll argue we've actually started to learn something, which I'll go over in my presentation today. So this is obviously a picture we've seen, Hans Berger, and I'm not going to focus on the history, which we've all heard about, the equipment, and of course the famous first EEG recordings. What I do want to draw attention to is that these recordings, they were obviously done by ink or a pen and paper at first, and before they were being shown on some computer screen. And the clinicians actually started looking at the waveforms and tried to understand what the waveforms tell us. And so, of course, Hans Berger demonstrated the eyes open, eyes closed relationship, as we've just heard about. And sometime later, in the 50s, we started learning about sleep, and of course, many other conditions, epilepsy and things like that. But the point was, up to, or for quite some time, for the first several decades, the analysis of EEG was really primarily or exclusively by necessity, because there was no other method, was done by humans looking at the signal. And so they were actually looking at it and describing what the EEG looked like. So the motor-related mu rhythm over central areas, or motor-related areas, they were called the mu rhythm, the arch-shaped rhythm, or the wicked rhythm, and things like that, because that's what it looked like. They didn't say it's a sinusoid or something, because it doesn't look like a sinusoid. Well, then, of course, later in the 1960s, we started to have computers, and so, of course, a relatively obvious thing to do was to start using computers to analyze the EEG signals. And this is Walter Freeman right here. So one of the big people doing computer-based, or first people doing computer-based EEG analysis. He was one of the first to apply the FFT to the data. This is some example published in 1963. This is some sensory areas in the cat, I believe. You can see some peak at 40 hertz. And the point is, so obviously you're going to want to use the computers to make the EEG analysis more quantitative, more efficient. And so using the FFT or some spectral analysis, of course, seems to be relatively clear that this is something you would want to do, given the fact that the signal looks like, something that is repeating. So the use of the FFT was relatively a relatively obvious choice. This was then obviously started to apply by all kinds of scientists. So this is an example from Wolpa and McFarland. This is BCI-related work looking at the mu rhythm that you can see on the top when the person is not moving, and when the person is moving the hand in this case, you see this rhythm disappear. And when you look at the EEG and the spectrum of the EEG, just like we've seen before in the visual areas, we see a peak around 10 hertz that this then disappearing with movement. This is not the point I'm trying to make. The point I'm trying to make here is that we're starting to use EEG-based or computer-based analyses, which of course are very helpful, but we're really forgetting something here. We're forgetting that the second that we start to analyze data in a way, like with the Fourier transform, for example, we are introducing a lot of assumptions about what it is that we're measuring. So like, for example, the FFT, the way it's working, it's based on sinusoids. So it assumes that the signal is sinusoidal, which we know it isn't. So that has a lot of consequences. It has consequences. There are the peaks in the spectrum that we see, like around 10 hertz, but there may be other peaks at other frequencies that are really only there because the signal is not sinusoidal. And so the FFT shows us something at other frequencies so that it can describe the signal altogether. But these are, so this is really very problematic. The other thing that is that the FFT is assuming, it's assuming that the signal is zero mean. Now, if we have a signal that is, so basically, as you can see here, the top actually looks like sort of zero mean. This is the EEG, and it goes like that, and then sometimes it has higher amplitude, sometimes it has lower amplitude, and you can sort of imagine if it was like centered around zero. Then that's kind of okay. Well, but what if it isn't? What if it's biased? So what if it's when it's like small, it goes like this, and then when it's larger, it goes like that? Well, then the use of the FFT is really not very appropriate. And what we have seen is in particular, in more recent years, we've seen layering methods upon methods. So, for example, in electrocorticography, that I'll talk about in just a moment, it's become very popular to do things like phase amplitude coupling. So it's the relationship between two different frequencies. Of course, we can have relationships between two different locations and things like that. We've heard about this earlier, like coherence and things like that. Now, when we do that, and the more of this mathematics we are applying, the more assumptions are implicitly baked into the analysis. And without us really knowing what the EEG actually looks like. And I was really refreshed by the Brad Wojtek's talk, who was obviously talked about really coming up with new methods to quantitatively describe the EEG, which I think is really something that's very much necessary. Now, a lot of my work has focused on electrocorticography, which is really a beautiful way to see the EEG in more detail. And this is obviously something that Hans Berger wasn't able to do, but that's now more commonly possible in epilepsy and tumor, as part of epilepsy and tumor neurosurgery worldwide. So we now not only have scalp recorded EEG, we have subdural recordings like ECOG, electrocorticography that you see on the bottom left. On the top right, you see a diagram of stereo EEG and DBS recordings, and obviously we can also have at least some access to multi-unit recordings in humans. Now, these kind of ECOG recordings have generated quite some quite some insights lately over the past 10 years or so, or 10 or 15 years. This is an example of what it looks like for people that have never seen this before. It looks rather crude, but they are grids that look just like the ones on the right. So these are clinical ECOG grids that are being used again in prior to epilepsy or tumor neurosurgery, and they're being implanted directly on the surface of the brain. And these signals, they have very high spatial resolution. This is an example of a grid recording that we did about, I don't know, like 8 years ago or so. This is a grid with 250 electrodes, 3 millimeter spacing, 1 millimeter exposed. So this almost has like fMRI type resolution, but it also has very high temporal resolution, which of course fMRI doesn't have. So this is just an example of brain signals, ECOG signals related to visual motor task. The subject sees a visual stimulus and then has to respond with a button press. And what you see here is a very timely progression. This is a single trial of cortical activation, first in occipital areas, the green traces that you see here, then followed by this blue area, temporoparietal junction, and then up to the pink one in hand area motor cortex prior to the button press, and then followed after the button press by activity in the dark blue trace on the very top, which is in hand area, sensory areas of hand cortex. So ECOG has very high temporal resolution. Now, it can measure two signals. One is oscillatory activity, which we've heard about a lot. This is low frequency, around 10 hertz or 5 hertz or something. And the same you would see in scalp recorded EEG. And now this, what has been known for a long time, actually this is from Bishop 1932, who called it the cortical excitability cycle. And so this is actually from that paper where you can see an oscillation, an oscillatory cycle that cycles in this case at 200 milliseconds, so 5 times per second. And it was known relatively early that high amplitude means low cortical excitability, meaning the cortex cannot be excited very much. And low amplitudes means high excitability. Now this matches. Today we measure, we don't think about amplitude. Most people measure oscillatory activity by measuring oscillatory power, which is really the envelope, basically the peak to trough amplitude, or the phase, which is a whole other topic. I strongly believe that measuring things by power and phase doesn't really make much sense. And actually going back to just measuring the amplitude is much more closely related to the actual physiological process, but that's a whole topic in and of itself. There's several papers that we published on this topic, actually showing that using an appropriate measurement of amplitude gives much better prediction of cortical excitability than either the power or the phase. But this is one signal that we can see with electrocorticography, just like we can see it in a scalp recorded EEG, relating and what it really means physiological. It gives cortical excitability. How easy is it for an external stimulus or from some other brain area to excite a particular area in the brain? Now, the second signal that we can see, and that we can only see in electrocorticography and not in the EEG, is the broadband spectral change. Brad has also talked about this, citing important work from Kai Miller and others that is talking about how there's really a 1/f function in the EEG, and that's relating to or reflecting local cortical activation, the cortical excitation. It's the firing of units, which in this case, in this example, if at this particular place, a particular location, we measure 10 firings per second, we get this blue spectrum. And if we have units firing 40 spikes per second, then it basically increases amplitude across all frequencies in this way. So in summary, we're really talking about two signals, one relating to cortical excitability, and the other one to cortical excitation. Now, the way they modulate each other, or the way they interact, has been studied for quite some time. This is an example from 1956, in which it's a recording from the cortex. You can see a motor cortical neuron firing happily along. And at the time marked with VL, the scientists stimulated a particular thalamic nucleus that projects to that area in the cortex. So at that time at VL, and what you can see is that this, at that time, this generates a positive voltage change in the cortex, during which the cortical neuron stops firing. So this now means, and this is, I propose, the mechanism that is behind oscillatory activity. It's rhythmic volleys that arrive from different thalamic nuclei at the cortex, hyperpolarizing the cortical voltage, just like shown you in, here, but doing so repetitively. So you get this oscillatory activity that we can see in occipital areas or central areas or other areas relating to different functions. And at the time at which the voltage is high, cortical activity is lower. So this is why we always see a negative relationship between oscillatory power, for example, if that's how we measure it, oscillatory power going up, broadband activity going down, and vice versa. So this is essentially a way for the cortex to, for the brain, and for the thalamic areas in particular, to modulate the cortex. And the model that is really arriving, or sort of that we're arriving at from in this picture, is a model in which we have a patch of the cortex that's marked here in C, that's under inhibitory influence from subcortical areas, which make the cortical surface potential go up. At the same time, there's excitatory input from either from sensory stimuli or from other areas in the brain that are excitatory and that make the cortical voltage go down. And if basically the excitatory influence is higher than the inhibitory influence, we get then this patch of cortex, the neurons in this patch of cortex will fire. So that's a very, very simple way of thinking about the cortex. Now, this is not completely proven, but I think there's very strong evidence that suggests that this way of thinking is correct. And it provides a direct link from individual cortical physiology of individual neurons under excitatory and inhibitory influence, up all the way to the actual measurement that we can do with ECOG or with EEG. And so just a few words on how the brain is actually using this. So, and maybe I just focus on the right side here. If you imagine different cortical areas, each labeled circle here refers to one area in the brain, and the arrows show how they are all connected. And if the brain wants to gate information from a stimulus, for example, to a certain target, let's say motor cortex, it can increase low frequency activity in all the gray areas, decrease this basically inhibitory influence in all the red areas. And so if a stimulus now comes in, it excites this area in A, action potentials are flying to all these other areas that are connected to it. But only at the location marked in C that has low amplitude oscillatory activity will it actually make that the neurons in this area C fire, and so forth. And so I propose that that the brain is using low frequency activity to functionalize the brain to do different things, even though the actual physical connectivity are all stay the same. And so with this, I think we are now arriving at in an area where we can actually use, in particular, these invasive methods that give us access to the two signals, the broadband signal we just cannot see, unfortunately, in scalp recorded EEG, and actually use the information derived from it when we do some thinking and not just throw math at it, and we do some careful thinking, just like Brad was describing, really trying to understand the signal. We can actually derive at important insights into brain function. And this is really all I had to say today. So these Chinese characters mean xie xie, which means thank you very much for your attention.